So my name is Devin. I'm an emergency doctor from Sick Kids Hospital, and I also have a master's in computer science from U of T. And I trained under Anna Goldenberg and Mike Bruno, some of the folks that you may know, who are professors over at U of T in computer science. And with that has grown into the role of co-lead of research at T-CAIREM, co-lead of some of the AI initiatives at Sick Kids as well. And I'm the co-founder and CEO of a health tech company called Hero AI. And so doing a lot of AI research and really always inspired by a lot of the student work that is being done because they're just such amazing and creative ideas. And it looks like we've got about 45 plus growing people joining. And it's about 12:01. So let's just dive right into our agenda for the day. So we've got two incredible presentations. They'll each be about 20, 25 minutes, and then we'll do questions after each presentation. And so please feel free to chime in and ask lots of questions in the chat. I'll do my best to moderate, and we'll see if we can have great conversations after each presentation. Just some formalities to go through as well. I just want to remind everyone, so the T-CAIREM, which is the Temerity Center for AI Research and Education in Medicine at the University of Toronto, is made possible by this incredibly generous donation from the Temerity family. And so T-CAIREM is also an interdepartmental center that serves as really a focal point for collaboration among healthcare providers, trainees, researchers, computer scientists, engineers, and even industry coming together at our center. And really our goal is to transform health through AI. And so, by attending this event as well, just note that we are recording the event, and it will just be used so that those who miss it can take a look and watch back. So why don't we get into our first presentation? And so that's going to be by Fumiya, who is a master's student in the Interdisciplinary Oncology program at the BC Cancer Research Institute. And so the research that he'll be talking about is titled Large-Scale DNA Organization Identifies Aggressive Prostate Cancer in Low and Intermediate Risk Patients. And so what a really incredible topic. I'll pass it over to you and take it away. If you get to about 12:25, I might just politely let you know that we'll move over to questions, okay?
Okay, that sounds good.
Cool. Go for it.
Hey, thank you for the kind introduction. Hello, everyone. My name is Fumi, and as I was introduced, I'm a master's student in the Interdisciplinary Oncology program at UBC under the supervision of Dr. Martial Guillaud and Callum Macaulay. So today I'll be introducing a project titled Large-Scale DNA Organization Predicts Aggressive Prostate Cancer in Low and Intermediate Risk Radical Prostatectomy Patients, just to illustrate some of the work that we've been doing to incorporate AI into our research workflows. And we work out of the imaging unit in the Integrative Oncology Department at BC Cancer Research Center. And so at the Guillaud and Macaulay lab, we're interested in image processing techniques, which can provide fast and automated pipelines for things like early cancer detection and cancer prognoses. And we've previously established many pipelines that can analyze things like the architecture of tissues or structures of cell nuclei. And we've recently begun to adopt more deep learning and machine learning based methods into our analyses. So for instance, in this project, we studied prostate cancer prognoses using a combination of deep learning based image processing and also machine learning, supervised machine learning algorithms. And a more detailed overview of the work that we've been doing can be accessed through this website or QR code if you're interested. So I'll just leave that up there for a quick second. And our interest in prostate cancer stems from its large health burden on the Canadian population, and also its high potential for image analysis techniques to improve patient outlook. So prostate cancer is the most common cancer among Canadian men, and it's the second leading cause of cancer mortality. Currently, the gold standard for prostate cancer diagnosis is a visual inspection of prostate cord needle biopsies by a pathologist, and the patient is then assigned a risk category based on 3 factors. So serum levels of prostate-specific antigen, which is a protein secreted by the prostate, Gleason score, which is a grading method based on how differentiated or how normal the biopsy looks, and tumor stage, which is how far the tumor has progressed. Patients are stratified into low, intermediate, and high risk based on specific values of these factors. And although the specific numbers shown on the slide are not relevant, what is relevant here today is that these risk categories then inform the appropriate treatment modality. So for instance, active surveillance may be suitable for lower risk patients where invasive treatment is not very desirable. Clinicians may also opt for options such as radiation therapy, including brachytherapy or things like external beam radiation therapy, and surgical interventions such as radical prostatectomy, which is the complete removal of the prostate. So today in this presentation, we'll be focusing on radical prostatectomy patients. And the thing with prostate cancer therapeutics is that despite the curative intent, prostate cancer has quite high rates of what's called PSA-based recurrence, meaning that there are prostate cells that remain in the body and are continuing to proliferate and make the PSA protein. And so in low-risk patients after radical prostatectomy, roughly 15% of these patients will have PSA-based recurrence. For intermediate risk patients, that number is closer to 30%, and for high risk patients, up to 50% of these patients will eventually experience PSA-based recurrence. So PSA-based recurrence is an issue because it greatly increases the risk of metastatic disease and often results in chronic or incurable disease. However, adjuvant therapies using radiotherapy methods after surgical methods or androgen deprivation therapies have been shown to actually lower the risk of this PSA-based recurrence. So the question here is which patients will develop clinically relevant recurrence and which patients will actually benefit from this early stage adjuvant therapies. The approach we took to answering this question is utilizing quantitative pathology. And quantitative pathology, as we see it, has three aspects or what we call pillars. The first one is morphometry, which is studying the individual cells or nuclei and their structure. The second is global architecture, which is the spatial arrangement of cells. So previously I mentioned the Gleason score, which although it's not completely quantitative, it will fall under the global architecture category. And third is the cellular sociology, which is a combination of the morphometry and the global architecture, and where we study these interactions between specific cell types. Quantitative pathology has the potential to become a powerful auxiliary tool for pathologists. For instance, it may be able to provide recommendations on high-risk areas to pay attention to in the biopsy, or it may also be able to analyze subvisual patterns, providing an increased depth of pathological insights, which were previously not available. So such new information may help the pathologist build a stronger understanding of the patient's current disease state and how it's likely to progress. In this project specifically, we'll be focusing solely on the morphometry aspect of this quantitative pathology. So we're analyzing cell nuclei in this project, and the nuclear morphometry is described in what we call large-scale DNA organization features. And these are just quantitative measurements of the nucleus. And there's three types of these LDO features, as we call it. First is morphological features, which describe the shape and the size of the nuclei, and there's about 24 of these features. The next is photometric features, which describe the quantity of DNA in the nucleus, and we have about 18 features. So this describes things like the staining intensity or the optical density of the nuclei. And lastly, we have chromatin texture features, which describe the structural organization of DNA within the nucleus. And we're able to calculate these features because we use a DNA stoichiometric stain called fulganthionine. And what using a DNA stoichiometric stain does is it allows for the precise quantification of DNA at each pixel in the image. And therefore, we're able to quantify things like the chromatin texture or like how much DNA is present in the nucleus. And now that we've covered the LDO feature framework, this leads me to my hypothesis and the project objectives. So we hypothesize that large-scale DNA organization features that describe, as I mentioned, nuclear morphology, staining intensity, and chromatin texture can predict PSA-based recurrence in patients that have received radical prostatectomy as their treatment. So the objective of this overall project is twofold. So the first is to develop and optimize a pipeline to analyze nuclear morphometry in a digital slide to predict this PSA-based recurrence. And the second is to identify key high-level nuclear alterations associated with increased risk for poor clinical outcome. However, in this presentation, we're only going to be focusing on the first objective, which is to develop and optimize this digital slide analysis pipeline. This pipeline is developed using data provided by our collaborator, Dr. Miha Pukul, and the General Hospital of Celje in Slovenia. So in this data set, we have a total of 27 patients that have this PSA-based recurrence with 80 biopsy images, and we have 88 patients that are recurrence-free with a total of 215 biopsy images. So the overall data set we have to work with here is a total of 115 patients and 295 biopsies. So I'll just quickly go over the overview of this digital slide analysis pipeline before going into each, going in detail for each aspect. So there's 3 distinct phases for this analysis pipeline. And the first one is clinical sample acquisition. So this covers things like performing the prostate core needle biopsy, performing the Fulgenthine staining, and getting the images. The next phase is image processing. So this is where we perform something called spectral unmixing, which I will get into a little bit later, and deep learning segmentation of the individual nuclei. And lastly, we have the prediction framework, which uses a combination of unsupervised and supervised machine learning techniques to sort of predict or identify which nuclei are associated with this poor clinical outcome, this PSA-based recurrence. So taking a closer look at how the image is actually processed. So we're starting here with a biopsy that has been taken from a patient, paraffin embedded and formalin fixed, then stained with fulgenthionine. This image is then spectrally unmixed, meaning that the signal from the stain is isolated from other components in the image, such as diffraction noise or in some cases, other stains. So spectral unmixing is a separation technique of signals for different stains. So for example, in HAD, you can separate the hematoxylin signal from the eosin signal, things like that. And we can also separate out some of the noise that occurs in the image when taking the image scan. This unmixed image is then processed by the sequential unit or the SUNET, which was developed in-house. So the way this works is by utilizing 2 units in sequence, we can achieve instance level segmentation of the nuclei in these biopsy images. So we first load the image, and these are converted into tiles and then passed to the first unit. The first unit then labels the geometric centers of all the nuclei present in this image patch. And the coordinates of these geometric centers are noted, and then new tiles with the geometric centers at the center of the new tiles are generated, like so. And then after this, the second unit then segments the single nucleus in the center of the tile and are saved as 128 by 128 images, along with its binary mask in a custom format we've developed in-house called CMG. And this is what the biopsy image might look like after being processed by the SUNET model. So we can see just how the nuclei are individually segmented. And just one small thing I would like to note is that we can see in the bottom right corner of this biopsy image here that there are pixels within this frame that belong to 2 different objects. So that's one of the strengths of the sequential unit, is that we don't have to prematurely cut or define unnatural or irregular borders of the nuclei, just based on other techniques such as thresholding, which we've had to do in the past. And on the top right corner here, we have what these are what the individual nuclei look like after they're segmented. So we can look at all of these as data objects. And once we have these individual nuclei, we can, make about 114 nuclear measurements for each detected nucleus. So once the LDO features are available to us, utilizing a Gaussian mixture model, we generate 10 nuclear subtypes based on said features. And the reason we do this clustering is so that when we make comparisons in the future in downstream analysis pipelines, we want to be comparing cells of the same type, or if we're comparing cells of different types, we want to be intentional about that comparison. So for example, if we're comparing different kinds of epithelial nuclei, for example, like tumor epithelial nuclei to tumor epithelial nuclei, if we want to be comparing stromal nuclei to epithelial nuclei, we want to be very intentional about that rather than having one mixed data set of many different kinds of cells that we haven't been able to separate. So once we have these different clusters, we perform binary classification on each of the nuclei. So for each nuclear subtype that we've generated, we want to identify which of those nuclei are associated with recurrence. And we call these recurrence associated nuclei, or RAM. So using the LDO features as an input, we train random forest classifiers to predict whether that nucleus comes from a patient with recurrence or a recurrence-free patient. And for a given biopsy image, we summarize these nuclei-wise predictions by putting them into a proportion. So the proportion of that nucleus type that is predicted to be recurrence associated. So that was a little bit, well, it might be a little bit confusing at first. So after we've done the nuclei-wise prediction for a given image, we will have a total of 10 proportions because we have 10 nuclear subtypes. And a forward feature selection model reveals that nuclear subtype 4 and nuclear subtype 8 are the most discriminative of recurrence and recurrence-free patients. So using the proportions of recurrence-associated nuclei in cluster 4 and cluster 8, a linear discriminant analysis is then trained to label the biopsy as if it's, as associated with recurrence or if it's associated with non-recurrence. And for the patients, if more than 50% of the patient biopsies are predicted to be recurrence associated, then we say that that patient is likely to develop PSA-based recurrence in the future. So that's it for our digital slide analysis pipeline for now. I will like to say that this is a very kind of like a pre-alpha, like very initial draft of the analysis pipeline, but the results seem to be pretty good so far, which I'll be getting into now. So I'll be going in the same order as the analysis framework. So first, starting with the sequential unit segmentation performance. So just starting with the top left corner, that table shows the DICE scores for each of the 2 units. So the first unit we have is the geometric center detection, which has a DICE score of 44%. And we have the center nucleus segmentation, which has a DICE score of 76.67%. And just looking at the number 44% for the geometric center detection, this number may seem a little bit low, but for our purposes, what we want that unit to do is to detect the nuclei in the image patch. So if we take a look at the example test image here on the right, there are a total of 14 annotated nuclei, and this center detection unit is able to find 12 of those. It misses 2, and it also falsely identifies 2 other nuclei, but just in terms of nuclear detection, which is what we need, it does quite well. So overall, this sequential unit system does quite similar to a human annotator. Moving on into the clustering nuclear subtypes. So the nuclei shown here are representative examples of nuclei from each of the 10 generated clusters, and we can see a little bit of a pattern here. So the clustering was performed using all types of LDO features. So that's the morphological, the photometric, and the chromatin features. And just looking at it in terms of shape, we see that there aren't so many different types. For example, like cluster 0 and cluster 5 seem to have very similar shapes, even cluster 6. But because we used the photometric and the chromatin texture features as well, we see that the clustering algorithm is actually capturing differences in the photometric and the chromatin textures as well. And here we have an image of what a cluster 8 nucleus looks like and a cluster 4 nucleus looks like. So we see a very lobulated epithelial-like nucleus, which is what we might expect, as those are what the tumor nuclei look like. So this is just the predictions of all of, so each plot shown here are the classification results of the test biopsy images. The distribution of the right distribution of each of the subplots show the recurrence biopsy images, and the distribution on the left shows recurrence-free patient biopsy images. So in total, there are 40 biopsies in this test set, balanced equally with 20 from each of the non-recurrence and the recurrence patients. And we see another interesting trend here. So The proportion of recurrence-associated nuclei seems to be generally higher in the recurrence patients, which is what we expect. And getting into the specific biopsy predictions. So on the left here are, on the left there are the cluster 4 and the cluster 8 recurrence-associated nuclei predictions. And using these two proportions to train a linear discriminant analysis, we get the test performance as shown on the confusion matrix on the top right table here. So this linear discriminant analysis is able to correctly identify 19 of the 20 recurrence-free biopsy images and 14 of the 20 recurrence biopsy images. So that translates to a sensitivity and specificity of 70% and 95% respectively. And in terms of the patient predictions, the Kaplan-Meier survival curve is shown here on the left. 85 of the 88 recurrence-free patients were correctly identified, and 23 of the 27 recurrence patients were correctly identified. And this translates to a sensitivity and specificity of 85% and 96% respectively. So just going back and comparing to the current clinical standard. Now, although the Gleason score itself is not used as a predictor of recurrence, but we can sort of use it as a pseudo predictor just to compare the sensitivity and specificities. So in our data set, there are 14 of the 27 recurrence patients have a lower Gleason score of Gleason score 6, as shown in the right top table here. 13 of the 27 patients have a higher Gleason score of 7. And 73 of the 88 recurrence-free patients have a lower Gleason score of 6, and the remaining 15 have a higher Gleason score of 7. So it seems that there is some kind of trend there, but if we look in terms of sensitivity and specificity, it has a sensitivity of 17% and specificity of 52%. So when comparing to Gleason score, we see that our digital slide analysis pipeline is outperforming the Gleason score alone when used as a classifier. So that being said, there are two primary limitations to this study. So the first limitation is the small cohort size, especially with the number of recurrence patients. So we have 27 recurrence patients compared to 88 recurrence-free patients. So this makes it a little bit difficult to generalize to other recurrence patients, especially because there's a great deal of variability in prostate cancer patients. So for instance, the results achieved by this image analysis pipeline may work well for the patients that were in this cohort or have a similar type of cancer such as acinar adenocarcinoma, which is the really, I guess, the most frequent one, but it may not translate as much to patients that have other prostate cancers, such as small cell carcinoma or other variants of prostate cancer. The second limitation is that the classifiers in this pipeline are trained using biopsy images, and they weren't separated based on the patient. So that means if some of the patients if there were patient level variabilities in the data set, this variability might have been missed by the classifiers. And this again makes it difficult to generalize to patients outside of this specific cohort. And both of these limitations point to the need for an external cohort validation, which can show us how the current classifiers are likely to generalize to other patients. In terms of other future directions, I'm currently working on Objective 2, which is to identify key high-level nuclear alterations associated with increased risk for poor clinical outcome. So for instance, different alterations such as nuclear enlargement or lobulation and chromatin alterations may have different pathological effects, or they might be occurring at different stages of prostate cancer progression. And by characterizing this, we may be able to get a more precise characterization of the pathological state of the patient, and that will give us a better idea of how the patient's prostate cancer is likely to progress. Additionally, we'll be visiting the other two pillars of quantitative pathology, which is the global architecture and cellular sociology. So we already have a project going in the global architecture, which is just analyzing how the cells are spatially distributed within the tissue, and cellular sociology, which is looking at how the specific cells are interacting, will be sort of our final stage of this project. And with that, I'd like to move into acknowledgements. So this project was funded by the CRHR grant and completed in collaboration with the General Hospital Celje with radical prostatectomy data provided by Dr. Miha Pukul. I'd like to acknowledge my supervisors, Dr. Marcel Guillaud and Dr. Callum Macaulay, as well as everybody in the lab who have supported me in this project. And with that, I'm ready to take any questions.
Incredible presentation, for me. I really loved how you spent a lot of time explaining through the details of the pipeline. I mean, I'm a peds emerge doc, so I don't know that much about prostate cancer and the staging, but I feel like the way you explained it was incredibly helpful to then understanding the value proposition of the model you're building. So really kudos to that. There's 4 questions that I want to throw towards you in sort of next 4 minutes. So let's see if we can get through them. The
Okay.
first one I had was just a quick one. So you talked about something that was really interesting to me, this two-stepped UNET process, which I loved how you described then that you were able to get overlapping boundaries of segmentation, which is really clever. I may steal that for a project that I'm currently working on to try that out. So thank you for sharing that. You talked about the dice score, and like step 1 of the unit was 44% or 0.44, and the second step was in the 70s. For that second step in the 70s, did you ever try just doing like the single unit directly to that second step? And did you find that it wasn't as the dice score was actually lower? Or did you actually achieve higher dice scores by doing this two-step process? Like, is it actually necessary to do the two-step process?
So the two-step process, and it's more so for, I guess, downstream analysis. It allows us to separate the individual nuclei as, I guess, objects in terms of data. So that allows us to do the feature calculation or like the measurements for chromatin alterations and textures and things like that for each individual nucleus rather than patches of images. So I haven't looked at whether it would generate a higher or lower dice score, but um, I guess just in terms, I haven't done like a proper study for this, but
Yeah, that's okay. But basically,
just
you're saying
in
there's
terms
no
of
work to
what
doing it.
I've experienced, it's I guess it's just how the unit works. It's a little bit easier when you have one unit trying to segment all of the objects in an image patch. And when you try to limit that to only certain regions of what you're trying to segment, such as the center detection or the only segmenting the center nucleus, it is a little bit harder to get there. It does take a little bit more data, a little bit more training. So I guess this is a complete guess, but if we were to just have one unit, I think it would have a higher dice score, but that wouldn't necessarily accommodate our needs in the downstream analysis.
Yeah, that's really cool. The second question I had for you was around your end predictive model. And so do you have any thoughts around still having the binary classification model, but making it multimodal? So also putting in maybe the patient's age, or I wonder if there's risk factors from the clinical chart or features about that patient that supplement in with the pathology images that you're using for the classification prediction that may drive up some of the precision and the performance. Have you had any thoughts about that with your clinical collaborators?
Yeah, so the eventual goal is to accommodate all of those.
Yeah,
This was just sort of like the first pass. We were just giving the LDO features for each of the different clusters into the random forest just to see if we can find nuclei that are associated with recurrence on average. So it's not like we were looking for something very specific in those nuclei. And so I guess when we are going to tune this pipeline, we would be including some of the clinical features as well. So that is on our checklist, I guess.
yeah, amazing. Some questions in the chat. One was around, so besides the random forest model that you used as that binary classification tool, have you tried other methods like an XGBoost or SVM? Was that so? Basically, any other model architectures or approaches besides the random forest that you experimented with?
Not to a deep extent, I guess, but the other model that we tried was logistic regression, and it
Yeah.
sort of had similar. Random forest just did a little bit better. And at this stage, we haven't tried to fully optimize each of the binary classifiers in terms of the nuclei. We were just trying to see, is it possible to separate out what we loosely define as recurrence associated nuclei? So yeah, that would be one of the steps going forward, is taking a closer look at each of the clusters and some of the characteristics that are unique to each cluster, I guess.
Yeah, no, totally. So that sounds great. And usually once you've got your pipeline set up, it's not too difficult of a task to swap in different model approaches, like
Yeah.
sub N, random forest, sub N, XGBoost, and just see what happens as a little quick experiment. So that's really great. The last question I'll ask is around, so it's also in the chat. Were the Gleason scores taken from the actual pathology reports, or were the slides reviewed?
I
The team
believe
is
they were
separate research team.
taken directly from the pathology reports. So this data actually comes from a previous study that we did in our lab, which looked at the architectural component of the quantitative pathology, I guess, pillars. And that's when they did the actual Gleason scoring. So they did revise it once
Yeah.
during the study. But yeah, I believe they were working with pathologists as well during that time to get those Gleason scores.
Yeah, incredible. So an amazing, amazing presentation. Really, kudos to the work you're doing. If the team out in BC is looking for some way to collaborate with Toronto, T-CAIREM would be happy to try and help facilitate introductions or collaboration of some kind. I think this is really promising work and would really love to support you as you take it to the next step. So please reach out anytime and happy to help you navigate that. So congrats on a great presentation. There are a few more questions in the chat. So feel free to get in there and just to type in some of your responses as well. And we'll pivot over to our second presenter. So our second presenter is Vivian. So Vivian is a second-year PhD student in the Department of Medical Biophysics at the University of Toronto. And Vivian is going to present a project entitled Mapping the Neoantigen Landscape of Patient-Derived Organoids Using a Peptide-Based Large Language Model, which sounds incredibly innovative. I can't wait to hear what a peptide-based large language model is. So super excited for this. So take it away, Vivian.
Thanks so much. I'll just do a quick revision of that title, though. I've modified it to be transformer model instead of large language.
Okay, perfect. That's all good.
Thank you. So hi, everyone. I will be presenting on identifying neoantigens in patient-derived organoids. And I'm part of the Wang and He labs in the Department of Medical Biophysics. So I'll just go ahead and get started. So to start, the immune response is the body's first natural defense mechanism. The immune response is designed to recognize and eliminate foreign invaders such as viruses, bacteria, and other pathogens. And this complex system is comprised of various cells, tissues, and organs that work together to identify and neutralize these threats. However, its role actually extends also beyond just fighting infections. It also plays a really crucial role in detecting and destroying abnormal cells, including cancer cells. So cancer immunotherapy works by leveraging one's own immune system as a therapeutic avenue. A range of immunotherapies have already been approved, bringing a lot of significant clinical benefits to cancer patients. So for example, here we highlight immune checkpoint inhibitors, which work by blocking checkpoint proteins like PD-1 and CTLA-4, and that suppresses T cell activity. And so by releasing these quote unquote breaks, T cells can better attack cancer cells. There's also CAR T therapy, which involves engineering T cells to express antigen receptors, and that enables them to recognize and kill cancer cells. There's also more, but I don't think I have enough time to go through all of the different types of cancer immunotherapies. But the premise of this is that to achieve such precise killing effects, the immune system relies on specific markers called neoantigens. And so neoantigens are a subset of abnormal peptides that originate from genetic mutations within tumor cells. So more broadly, tumor-specific mutations will produce these novel protein sequences, which are coined as new or neo fragments of proteins, in our case termed antigens. And these peptides are typically 8 to 10 amino acids long, produced by the proteasome. And then these antigenic peptides can then associate with the major histocompatibility complex, or MHC class 1 molecules, in the endoplasmic reticulum. and then migrate to the cell membrane. And it's at the cell membrane where the activated CD8 plus T cells can recognize and then bind to these target cancer cells, waving their flags. And this process is a cycle in which the tumor cell death response results in the release of more and more of these antigens, and then ultimately, hopefully, improves the immune response to tumor cells. So therefore, the core of cancer immunotherapy using neoantigens is to activate the killing function of these T cells and also promote the presentation of neoantigens. So there are three main steps in the neoantigen immune process. First, there's binding with the major histocompatibility complex, which is also known as the human leukocyte antigen, or HLA, in humans. There's also extracellular presentation and induction of immunogenicity by forming interactions with the T cell receptor, or the TCR. And in recent years, a plethora of machine learning models have been developed to predict each of the above three processes related to neoantigens for binding, presentation, and immunogenicity, each of which contributes to neoantigen identification in very different ways. However, despite the process made in both scientific research and also clinical therapeutics, the overall accuracy of neoantigen identification remains far from satisfactory. And so that's why we think that this might be attributed to source, where neoantigens can arise from multiple levels of genomic, transcriptomic, and also proteomic mechanisms, including just normal single nucleotide variants, different types of splicing, and also post-translational modifications. So there's a lot of layers of abstraction here. And over the last decade, most of the focus of neoantigen sources have been focused on somatic mutations derived from these single nucleotide variants, or SNVs, and also small insertion or deletion, also known as indels, that accumulate during cancer progression, which can also be measured by tumor mutational burden. A limitation with these SNV neoantigens is that they are patient-specific, and their accurate prediction is critical for the development of these effective immunotherapies. And so tumor mutational burden might not be sufficient to mount an effective anti-tumor response in many cancers. Going a little bit more in depth of these SNV predicted neoantigens, here in a meta-analysis of about 13 different publications, it was actually found that the number of SNV predicted neoantigens reported to be immunogenic was as low as 3%. So you can see they're very low numbers compared to the no in this column. Therefore, key determinants of neoantigen immunogenicity is not yet fully understood and incorporated into these neoantigen prediction models. So I think that alternative sources of neoantigens, such as gene fusions, alternative splicing variants, have emerged as attractive targets for immunotherapy. And I'd like to focus on alternative splicing in particular. Alternative splicing is an essential process for the maturation of nearly all mammalian mRNAs, where pre-mRNA introns are removed and various exons are selected and joined together, creating really diverse transcripts and proteins from the same DNA template. In fact, SNVs and indels also highly contribute to alternative splicing as well. And this is particularly relevant as splicing dysregulation is detected in many cancers. And in this plot, we have the x-axis as various cancer types and the y-axis as the frequency of these exon skipping events. And so we can see from, I believe this was a study of 8,705 patients, there's a lot of differences in alternative splicing patterns. And lastly, to top it all off, neoantigen-directed therapy also lacks a lot of preclinical models which accurately recapitulate the neoantigen characteristics of original tumors. So typically, sequencing data is actually extracted from cancer cell lines. So one method to address this is to use patient-derived organoids. For those who don't know, organoids are three-dimensional cell aggregates with near physiological cell behaviors and can undergo long-term expansion in vitro. Using these organoids, we can screen a large number of computationally predicted peptides. So therefore, I hypothesize that expanding the search for neoantigens beyond traditional SNVs and indels to include the alternative molecular alterations and also using PDOs in combination with some interpretability methods will result in a more accurate and comprehensive profiling of these neoantigens. So my research aims are as follows. First, I will identify alternative splicing neoantigens from these PDO RNA sequencing data. And then my aim too is to develop a machine learning model to predict these immunogenic neoantigens. And that leads us to my first aim. So for our data source, the Princess Margaret Living Biobank is a comprehensive resource for cancer research. It's located at the Princess Margaret Cancer Center in Toronto. The biobank consists of patient-derived cancer models, including organoids and also patient-derived xenografts, which are created from tumor samples obtained from patients undergoing treatment at the center. We're also, so from this biobank, we have obtained whole exome, whole genome, and RNA sequencing from the repository, and with a pan-cancer approach using pancreas, colorectal, lung, esophageal, ovarian, breast. In total, we have RNA-seq obtained from 162 PDOs. So to identify alternative splicing events, my workflow is as follows. First, I take the raw RNA-seq FASTQs and run a quality check, and then trim low-quality reads if necessary. Then, taking the trimmed FASTQs, I use STAR to build and map reads to a reference genome. In this case, our reference genome is HG38, and we obtain our transcriptome mappings. Thirdly, we take the transcriptome mapping and also the reference annotation. Again, it was HG38, and we quantify the reads contained in our samples to obtain transcript annotations, which contain information about intron exon regions and also the raw sequence itself. And lastly, I identify alternative splicing events by comparing the transcript annotation, again, with the reference annotation. And then using this tool called GFF compare, we filter for high quality and high confidence events and then we obtain our list of alternative splice transcripts as sequences alone. So this is a multi-FASTQ file. But in terms of alternative splicing events, we want to identify novel ones, right? So we can observe whether the transcript has an exact match to exons in our reference, whether a exon partially covers an intron, or whether the transcript actually retains all introns. In my pipeline, I filter for any transcripts which might be unique and not found exactly in the reference. Remember, we want to identify unique and mutated tumor-specific peptides. And so from this, we've identified at least 3,000 novel alternative splicing annotations from per sample in our tumor types. Here we can reference the total novel junction and unknown annotations for each specific tumor type. This is a sum of all of the annotations in total. You might be asking why I might have summed it. There might be a lot of redundancies. Well, I've reasoned that in my deep learning pipeline, I want as much data as possible and as many sequences as possible to be able to predict whether there is a neoantigen event that might occur within that sequence. And these are not all redundant per se. There might be one or two nucleotide changes within these transcripts. And yeah, so this is a graph also showing the same information. We've identified at least 3,000 novel alternative splicing events per sample within each tumor type. And ovarian has a lot of these annotations. If you ask me why, I'm not exactly sure. It might just be an imbalance within the data itself. Following annotation, I next aim to perform HLA typing on our RNA-seq to generate pairs of peptides and HLA sequences using this tool called ARCUS HLA. My next aim is to develop a machine learning model to predict immunogenic neoantigens. So existing algorithms are actually trained on a limited set of peptide HLA sequences, and we also want to know how these models arise at their predictions. So remember this slide I shared before. There's 3 steps in the neoantigen immune process. There's binding, presentation, and response. For measuring response itself, response can be measured using MHC binding assays. So whether there is a high affinity for peptides to bind to these MHC molecules. And there's also cell-based assays. So we evaluate the ability of these already formed peptide MHC complexes to stimulate an immune response, particularly with T cells. So these would be like T cell activation assays or tetramer staining. From the immune epitope database, I've obtained 500,000 unique peptide sequences and 151 MHC1 sequences. These sequences are obtained from T cell, B cell, and MHC ligand assays, and the binding between the peptide and MHCs are measured, as I mentioned before, using those assays. To this end, we've developed a transformer model to predict neoantigen immunogenicity. So this uses a Siamese transformer, which is a modification of the transformer model. This architecture is typically used in tasks that require comparing or finding relationships between two inputs. So it would consist of two identical subnetworks that share the same weights and parameters. So just to walk you through the architecture, each input sequence, so this would be the MHC peptide sequence or the peptide sequence itself, is passed through an embedding layer, and then we convert those tokens into dense vectors. And we tokenize this using an existing model called ProtBERT. ProtBERT is a BERT-based model specifically trained on proteins. Then the encoded inputs are passed through a series of transformer encoder layers, and since the weights are shared, the same transformations are applied to both inputs. Then the transformer encoder extracts the contextualized features from both the input sequence of MHC and peptide, and we can, I then perform a similarity function to see how similar the two sequences are to each other. We then concatenate the outputs and pass it through an additional layer, which is this MLP layer, to learn the relationship of the two inputs. And the output of this is binding affinity, measured as a score of 0 or 1, whether it binds or it doesn't bind. And so this is the performance of the model. So a couple of experiments were performed. So initially we experimented with a single model, which is just one transformer encoder block. So concatenation would occur first before we pass it through. And it performs probably not as well. And then we also compared it to another tool called MHC AttentionNet. We also experimented with pre-training only, sharing the weights only, and then also combining weights and pre-training. And combining the weights and pre-training in this Siamese transformer model architecture. So this is what I referenced for double. So double the transformer encoders performs the best. In addition to performance, we also see that increasing maximum sequence length improves performance. And we can see this over the loss over the training steps and also the AUC-RC over training steps. So the orange references the maximum length of 128 nucleotides, and then the red is maximum length of 32 nucleotides in the sequence. So my next steps are to benchmark performance with existing neoantigen identification models and also incorporate TCR sequence with the third transformer block. And yes, so that is all for my presentation. In summary, we've identified alternative splicing events in the PDO-RNA-seq and also developed a deep learning model to predict binding affinity among the peptides and their HLA or MHC tags. And here below, I have kind of summarize it into a nice infographic. First, we have the organoid RNA-seq. We perform quality check, alignment, and quantification, identify the alternative splicing events, and then pass those alternative splicing events into our neoantigen detection pipeline. For future directions, we would love to experimentally validate these neoantigen candidates in vitro using cell lines, and also conduct some interpretability analyses on the PEP-MHC model. And with that, I'd like to acknowledge and thank all members of the Wang and He Labs, and also members of the Princess Margaret Living Biobank, and my supervisors, Dr. Bo Wang and Hanson He. Thank you.
Amazing, incredible work. And I just want to shout out like both of our trainees. These are really complicated stories to tee up, right? Like some of my work is so basic. It's like, does a kid have appendicitis? Yes, no. Like it's so easy to explain that problem setup. But like for both of these presentations we saw, and for some of the trainees out there, the way that these two really set up their pipeline, explain the problem, even your use of graphics, Vivian, on how you went like one piece and then it went over to the other and the box kind of floated up, really gentle animations, but it was really, really helpful with understanding what is an incredibly complicated pipeline. So really congrats on doing that. I did have a few questions, and then we'll see if any other questions pop up into the chat. Can you go back to the slide where you showed like the two types of inputs going through the transformer models, the concatenation, one more, but yeah, this one here. Can you just explain a little bit more what's happening at the MLP stage here? I might have missed it.
Yes, so the MLP is a binary classification layer. Just to walk you through again, we have our MHC sequence, which is the nucleotide sequence in itself, sorry, the peptide sequence in itself. Then we have the peptide sequence, and that's encoded in the transformer encoder, and then we concatenate those vectors together. So it would be MHC and then just following the peptide sequence. And then the MLP is there to perform binary classification. So whether it binds or it doesn't bind, and we have that information from the IEDB database from all of the assays.
Yeah, and so what type of model are you using there to actually perform that final classification task?
Yeah, it's just a linear layer.
It's just a, say it again.
It's a linear layer.
Yeah, okay.
It's a linear layer.
It's pretty simple. All the heavy work and lifting is happening upstream.
Yes,
as you
exactly.
showed it. And then if you go back to then your performance charts here, it looks like you outperformed. So this like bolded model at the bottom here, so the double weights plus pre-training, is this sort of your final model attempt and your final approach attempt, and those are the results that you achieved? Or can you walk us through a bit here?
Yeah, sure. So the single model is the one transformer encoder block. And then the double would be the two transformer encoder blocks, one dedicated to each, either the MHC sequence or the peptide sequence. Pre-training refers to initial pre-training with the 500,000 peptide sequences that I mentioned from the IEDB. Weights would refer to extracting the weights from the prot-BERT model, which is the BERT-based protein model. And then the double would be combining both the prot-BERT weights, which we reasoned prot-BERT has already learned the semantics of what a protein sequence is, right? And then pre-training would be combining that. And to your question of whether this is our final model, I don't think there ever can be a final model. There again, we have 5 epochs that were run. Ideally, we would like to run more. In addition, we'd like to incorporate some clinical data as well. We are working with the Princess Margaret Living Biobank, so they have information about age, kind of like onset and whatnot. So I think it can definitely be improved.
But what's, I just wanted to underscore though, so like this by final approach, I guess I mean the final approach in the context of your presentation here, it looks like that really outperformed the MHC attention net.
Yes.
Here.
Yes, in the context of the presentation, yes.
Yeah, and even like this sensitivity you've got in green under the MHC attention net of like 0.9277, really is that just a manifestation of the decision threshold that you set, right? Because you look here, your AURC is outperforming, the PRC is outperforming, and so really the sensitivity potentially depending on where you've got the decision threshold for how the model's operating might go up or down, right?
Yes, exactly.
Yeah, so you might be outperforming on all parameters there, which is really, really cool to see how you've done that. A question has popped up into the chat, so I'll just read it. Can you elaborate on the evaluation metrics used on the ProBERT model and how it informs the performance with respect to omics data? So in brackets, since most evaluation metrics on BERT-like models are used for NLP tests only. Thank you for the presentation. So that any feedback on that?
I'm not sure if I can answer this question to like as detailed, but I do believe that ProBERT uses the MLM or the mass language model objective. So it would just be comparing how well ProBERT would kind of match up or create or fill in those like mass sequence areas of those proteins. Just a simple whether it does align or it doesn't align.
Yeah, amazing. Just we have a few other questions. I noticed, so the initial slide had said large language model and then transformer model. And I know that transformer models and large language models often, like in my research, I'm looking at thinking, well, can I actually replace this transformer model that's a little bit more cumbersome to use with a large language model? And I'm curious, like, was that a bit of a hint around another approach that you might be looking at that you feel comfortable with sharing? And if not, that's okay, but I'm just sort of digging in out of my own curiosities.
In regards to the large language model portion, I think that was a blip in my own when I submitted the project. But yes, the overall architecture, that is at least like what we've built here, is the transformer model.
Yeah, amazing. Okay, I was just curious. And so you're getting lots of compliments in the chat as well, so check those out. But maybe what we'll do at this time is just sort of everyone sort of put your hands together for our two trainees, two incredible presentations. And if you're a trainee in the audience or the two of our presenters here from your research labs, feel free to reach out to T-CAIREM in any way. We're looking to foster and grow our community through collaboration with this type of research. And so these are two incredible presentations that we'd love to see get taken to the next step. So congratulations on all the incredible work. Thank you to everyone who came out and spent a bit of their summer with T-CAIREM today. And look forward to seeing everyone at the next trainee rounds. I'm just gonna say a quick reminder that we're holding our final trainee rounds this summer on July 23rd. And so take a look out for that, register and see everyone then. This is incredible work. Thank you to our trainees for all the effort. Bye for now.